So my name is Mikayla Borton, and today I'm going to be talking to you about my work at Colorado State University to inventory river microbiomes across North America. So switching gears a little bit from humans, rivers are an important connection between terrestrial and marine ecosystems. And so what this figure is showing is the connection on how that connection might impact their microbial community. So rivers flow from the headwaters or smaller order streams down to larger order streams with inputs from across the land within that flow path. And so you can imagine things like land use, along with their additive effects, might have impacts on microbial community function. And so these microbiomes are critical for roles including things like fixing CO2, processing waste removal, as well as greenhouse gas production. And so despite these microbial communities' importance in these ecosystems, up until the project I'm going to be talking to you today about, microbial and viral studies, particularly biogeography studies from river systems, were scarce. So specifically those that focused on a genome resolved view. And so we had a lot of sequencing information about a specific set of rivers, but they were not biogeographically distributed. And so this figure that I'm showing you is showing you the number of biogeography studies per type of ecosystem in the last few decades. And so you can see that inland water bodies in light blue are relatively small compared to their soil and ocean counterparts. And so we catalyzed a community sequencing effort to address this gap, and that is called GROW, so the Genome Resolved Open Watersheds database. And so this was a community effort to sequence global rivers. And so what this map is showing us is all the locations of GROW. And so each point is a sampling location, and the size of the point is the number of samples collected at that site. And then the points are colored by the project within GROW, which I'll talk about some of the projects coming up, the projects within GROW that contributed those samples. And so you can imagine this is a lot of sequencing and a lot of sampling. And so this effort couldn't have been done alone. And so we teamed up with 19 other scientists to sample our global rivers. And so that is what is shown in this network here. And really, I want to highlight that none of the people on this screen were united by a single source of grant funding. What really united all of us was that we teamed together to get a joint Genome Institute community science project. And so that was led by Kelly Wrighton out of Colorado State. And that was really to just fund the sequencing. So then individual PIs were able to contribute different samples from their projects to sort of put into GROW and start to collate all of this information. And so one effort I really want to highlight here, given the topic of this seminar, is WONDERS. And so WONDERS is the Worldwide Hydrobiogeochemistry Observation Network for Dynamic River Systems. And so that's the data I'm mostly going to talk to you today about. But this was truly a community effort to sequence world rivers, where we sent out sequencing kits or sampling kits. And so you can see a picture of a sampling kit here. So WONDERS provided stream sampling kits to anyone who was interested, researchers who were interested throughout the world, to collect surface water and sediment. These were curated to collect several types of data, so metabolomics data in the form of FTICR, basic geochemical analysis, and then the microbiome samples. And so each of these data types went through different streams. All the samples were shipped back to Pacific Northwest National Lab, and then the microbiome samples came to Colorado State, and then the other data types stayed at PNNL for them to do the metabolomics and as well as characterize the geochemistry. So all the funding for the kits and the chemistry was provided by the U.S. Department of Energy. So this resulted in a very large data resource. And so these are all the types of data that were collected. So we have, this number is continuously growing because we're continuously submitting samples, but we have over 800 metagenomes now, all sequenced at the JGI. We were able to pair that to geospatial information. So for every location, we were able to pull different things like land use, climate data for that location. We also got FTICR data, geochemistry, and then metatranscriptomes, where we were able to get enough RNA. And so the sample, I'm going to tell you a little bit of a science story, because as I said, prior to this work within GROW, there was really no biogeography study of rivers where we could compare across. And so the subset of samples I'm going to be talking to you today about is surface water microbiomes in the United States, and they were all collected and processed using consistent methods. And so from a subset of this data, about 150 samples, we were able to recover over 2,000 unique metagenome assembled genomes. And these were from 27 phyla, with a majority of the genomes coming from proteobacteria, actinobacteria, and bacterodota, which is consistent with previous 16S studies in these systems. We were then able to map our microbial expression to this database to understand the microbial activity in these ecosystems. And we found that 25% of genomes in GROWdb were expressed in at least in the metatranscriptomes. And so, as I said, we were really interested in tying the biogeography data to the microbial community data. And we were also interested in the types of functions that we could see across these different spatial and geochemical gradients. So we collated watershed characteristics and land use for every site, as well as geochemical information. And that was by teaming up with the geospatial centroid at Colorado State. And so our first question was, at a bulk level, do microbial communities relate to these river features across such a big geographical distance? And so one feature that we were particularly interested in was stream order. And that is because it gives us a proxy for the size of the river. So a first order stream is small, which you can think of something like Como Creek, shown here for reference, while stream order 10 is something like the Mississippi River. So this is a large order stream. And so we hypothesized that this factor in particular would explain a large portion of the microbial community structuring, because it's directly related to the amount of inputs on the microbial community. We found that stream order structured microbial communities across rivers. And so you're looking at an NMDS here, and each point is a microbial community, and they're colored by stream order. So you can see the color gradient shows that microbial communities are more similar from smaller order streams and different from microbial communities of larger order streams. But of course, we also wanted to know, how does microbial community function change across stream orders? And so we were able to do this because we had all the metagenomic data to ask this question. And so we annotated GROWdb with DRAM, so distilled and refined annotation of metabolism, to understand the different carbon dynamics. And what we found was pretty cool, that we could see use of complex carbon significantly decreases with stream order, while smaller carbons, things like C1 compounds, increases across stream orders. And so we were able to ask new kinds of questions given the functionality information within the metagenomes and the metatranscriptomes, but also pairing it to these large biogeographic data sets. Because this resource was generated by the community, one thing that was very important to our team was making this resource accessible. So I'm going to talk about a few ways that we did that, because we tried to do it at different levels of data use. We did make all of the data publicly available as soon as it was sequenced within the JGI platform, but then we took it a few steps farther. So one way that we did with this, there were multiple data streams that I talked about. So the geochemistry data, the metabolomics data, the metagenome and metatranscriptome data. These are multiple data sources that are most of the time stored on separate platforms. And so the National Microbiome Data Collaborative, NMDC, allowed for linkages of all these data types across platforms. And so you can link out to all of the data through the NMDC. We also teamed up with KBase. So KBase is a platform that is publicly available, and you're actually able to, users are able to input their own data, but also utilize the GROW data to analyze in whatever way they see fit. So all of the microbiome tools, assemblers, and reed trimming and DRAM are all publicly available within KBase. And so users can go in and interpret the GROW data for themselves without having, maybe they don't have access to certain types of computational infrastructure. And so this makes it usable beyond just database files. We also generated a GUI interface for interacting with the data types, regardless of your scientific background. So this has all of the data at higher levels where users can go explore the data in terms of, I want to know, what certain microbiome function is correlated with stream order or other types of geochemistry data. And so we were able to make that possible by teaming up with the Geospatial Centroid. And then lastly, based on GROW data, we have been able to put forth several education and community-based research publications. So one example of that is we used the data for a series of crowdsourced publications. So actually, we set up different narratives with the GROW data, and folks could join and write and interpret the data. And those resulted in 5 different publications that were tied together in a Frontiers in Water research topic. We were also able to use the GROW data in workshops for International Society for Microbial Ecology. So this is the one we did last year. We are doing one this year at ISME Latin, and that leverages GROW data from that region. So for Cape Town, it was a Congo River data set that participants were able to attend the workshop and then subsequently lead a publication together in collaboration with KBase platform to publish that data. And then with that, I'd like to thank all my people on my team, specifically Kelly Wrighton and James Stegan, have